fructokinase? Six carbon. After this step, you break down your 6 carbon to the 2, 3 carbohydrates, like glyceraldehyde, 3-phosphate, and dihydroxyacetylphosphate by the help of aldolase. As you can see here, this is a reversible and it is not regulatory. After some isomerization, again because this is a ketosis, it is an aldosis. This form convert to this form. Please pay attention. Dihydroxyacetone cannot continue the pathway at first. It should convert to the glyceraldehyde, and now glycolysis continue. Yes.
The previous question was choose the regulator enzyme of what?
Glycolysis pathways. The major regulatory enzyme in a glycolysis pathway is phosphofructokinase. Okay?
Hexakinase also can regulate.
Hexokinase, yes. As I mentioned, we have 3 regulatory: hexokinase, phosphofructokinase, and the last one is the last one enzyme. I didn't show it yet. Pyruvate kinase, okay? The last reaction actually considered as a regulatory. So, this is the overview. As you can see here, 6 carbon stage, glucose 6 carbon, 6 carbon. Finally, at the end of first stage, you have a 3 carbon. And as I mentioned, you require energy in the form of ATP. This is the overview. 2 ATP is required, and actually 3 carbon carbohydrate produced. What about this second 5 reaction? Please consider this point. In this slide, actually, I didn't write the number of the substrate and product, okay? But you should consider that this reaction repeat twice, okay? Because why? Because at this step, if you remember, you have 2 glyceraldehyde, okay? So you should multiply all of them, okay? Multiply 2. Whenever glyceraldehyde 3-phosphate enter to the second 5 reaction, following to 1 dehydrogenase enzyme, and after adding inorganic phosphate, NADH is produced. But as I mentioned, because you have 2 glyceraldehyde, I wrote 2 here. Clear?
Yes.
And this is an irregulatory enzyme actually because this is a reversible. And after that, 1 and 3, bis phosphoglycerate. What is the difference between bis and di, like adenosine diphosphate?
Doctor, bi means the 2 phosphates have attachment to 2 adjacent carbon.
Or different?
For example, carbon number 2 at carbon number 3. But bis means the 2 phosphate attached to 2 carbon, but these 2 carbon have a carbon between them. For example, carbon number 3 and carbon number 4.
About di? About
Bis.
bis. What about di?
Di, 2 adjacent carbon.
Exactly bind to the one side, you know? For example, ADP. Whenever you have the ADP, for example, this is the, I don't know, adenine, and this is the ribose. They bind, for example, number 1 phosphate, number 2 phosphate. When we say ADP, both of the phosphate bind to the same site, okay? But when we say bis, it means that 1 and 3 bis, 1 and 2 bis, okay? We cannot say 1 and 1 bis. 1 and 1 means di, okay? At the same side, we have a di. Okay. After conversion of 1 and 3 bisphosphoglycerate by the kinase ADP, convert to the 2 ATP, and then 3 phosphoglycerate release. So the carbon, the phosphorus position of the 1 used for the conversion of the ADP to the ATP. So 3 is remain the location of 3 turn to the 2 position. And after that, by enolase, by the release of 1 water molecule, the molecule known as a PEP. Please remember this abbreviation because in the next slide I use it a lot. Okay? PEP. PEP is the abbreviation of phosphoenol pyruvate. Okay? And at the next step, as you can see here, this is a irreversible. So this molecule is another regulatory enzyme. How many maybe I want to review some sample questions. How many NADH produce during the second 5 stage? 2 NADH. Please pay attention. It's better to put this one. All of these are 2. Okay, I didn't know about these 2, but for your memorize it better, I put the right number on 2. These are correct number. When I wrote these 2, I consider that we have a 2 glyceraldehyde, okay? So during second 5 reaction, 2 NADH is produced, okay? Clear? I didn't draw 2 from, for example, 2,3 phosphoglycerate. I didn't draw it because it makes a busy slide, okay? But because the amount of energy is important, I just wrote the number 2, okay? So if I ask you from 1 glucose, okay? From 1 glucose to pyruvate, how many NADH is produced? 2. And 4? No. How many ATP produced? Let's pay attention to different question. How many ATP produced during the second 5 reaction? 4. 1, 2, 3, 4. How many ATP produced from glucose to pyruvate? 6. We cannot say. Please wait, I will tell you how we can calculate.
One NADH, 3 ATP.
Yes. Yes.
Victor 7.
It is complicated and we cannot say it is very easily. What is the question? Aerobic and anaerobic, okay? Some please pay attention. Maybe I want to confuse you, okay? And I ask you in an aerobic, anaerobic. In a first step, in a actually second step. At first you should imagine what is the situation. Is it aerobic or anaerobic, okay? Question, do you have a question?
Total amount is 7 ATP.
7, why?
Because
Please wait, I will show and calculate. Now, okay, wait. I will show the slide how we can calculate it, okay? At first, it's better to have a review on the main importance of the second point. As you can see here, as I mentioned, whenever you have a phosphor in the molecule, it means that it has a high energy. So, the red line are high energetic molecule, high energy molecule, high energy molecule, okay? So they produce it ATP, as I mentioned, because we have a 2 glyceraldehyde, 2 ATP here, 2 ATP here. The total gain of energy from the glycolysis, from the second half of glycolysis is 4 ATP. But you should know that we can get the energy from NADH. Okay, I will show you how we can calculate this one. Before that, I want to talk about the difference between hexokinase and glucokinase. Please pay attention to this here. If you remember remember, do you remember the Km? What is Km of enzyme? The concentration of substrate at which we have a half Vmax. Do you remember the concentration? For example, if the Vmax, for example, here is 100, the half Vmax is 50. Am I right?
Yes.
And this is the substrate concentration at which the Vmax is half. Clear? Do you understand? And if you remember, Km show the affinity of the enzyme for a substrate. Okay? When I have a high Km, what does it mean?
High affinity.
No.
Low affinity.
Low affinity, okay? They are actually opposite to each other. So when I have a high Km, low affinity, like this one. Please tell me, hexokinase and glucokinase, both of them has the same substrate, and the substrate is glucose. I want to ask a question. I want to actually review by some example question. Between glucokinase, the red curve, and between hexokinase, blue curve, which one has a higher affinity to substrate?
Hexokinase. Hexokinase.
Why?
It's because a low concentration of hexokinase have half Vmax.
Okay, yes. As you can see here, hexokinase has a very, very low amount of actually Km. Means that it's low glucose. Okay? So, what does it mean, as I mentioned, as I wrote here, hexokinase has a low Km? It means that this low Km show the high affinity for glucose. For example, imagine that we are in Ramadan, okay? At the near of Azan, okay? Maghrib, I don't know what you say. The amount of glucose is very, very low, okay? But we are alive, we are not dead, okay? So we require energy, or better word, we require glycolysis for the brain, okay? So whenever the amount of glucose is very low, exactly like Ramadan, okay? Near the iftar, okay? It means that even a small amount of glucose that remain can phosphorylated by the hexokinase, and glycolysis pathway continue to produce the energy. Do you understand? Okay? So, what is the importance of hexokinase? It is important at first in all tissue, but then especially whenever you have a low, very low concentration of the glucose, it can phosphorylate glucose to glucose 6-phosphate and continue glycolysis to produce energy, especially for the brain, to prevent a faint. Okay? If you have a zero glucose, you faint. Absolutely. Okay? Do you know what's a faint?
Yes. They lose consciousness.
What the answer of which one
About, please tell me. As I mentioned, please pay attention. I want to repeat my question. As I mentioned, Km show the affinity, okay? But there is an indirect Km affinity. So whenever Km is high, affinity is low. Then Km low, affinity is high. So according to this fact, you please tell me which one has a higher affinity on you between glucokinase and hexokinase. Which one has a higher affinity? As I mentioned, higher affinity and Km. has a lower Km. Please show me, please pay attention. Here. For example, I want to help you. Please pay attention to me. For hexokinase, for example, Km is 1. For glucokinase, Km is 10. Which one is higher? Km, this one is higher. What about affinity? Okay, clear? Do you understand? Okay, so this is the importance of the hexokinase. What about glucokinase? Glucokinase, as you can see here, has a different curve. Km is high. It means that in a normal, whenever the amount of the glucose is not very high, it is at the rest, okay? For example, I'm glucokinase. You have a daily, for example, activity, and you didn't receive any cake or chocolate, okay? I'm in a rest position, okay? Glucokinase, okay? But whenever you eat one fruit juice or one delicious creamy cake, I want it now. Okay? Then you eat it, suddenly it increases. Oh my God, the concentration of glucose is very high. So I want to start my activity. So for what? Because I want to decrease the blood sugar. Okay? How it can actually decrease? You should at first consider the tissue distribution. Glucokinase, if you remember, hexokinase, in all parts of the body present, hexokinase. But glucokinase, 2 organs. What are them? Liver and pancreas. What about the pancreas? What does it do? What is the function? It means that whenever you eat this delicious cake, it's reached to the pancreas. Pancreas sends the high glucose, okay, and secreted insulin, okay. And not only secreted insulin, it accelerates the activity of the glucokinase to all of the glucose, convert to glucose 6-phosphate, and actually they remove the glucose, okay, to control the level of blood sugar. Clear for pancreas? And about liver, exactly like a pancreas. It can, whenever you eat a chocolate, it enters from intestine, it enters the bloodstream, and from portal vein, it reaches to the capillary liver. Now, capillary sense it, high glucose, and start and wake up the glucokinase. And actually, glucokinase start the conversion of glucose to glucose 6-phosphate to fructose 6-phosphate, and glycolysis continue. So whenever, in other words, whenever you have the active glycolysis, you can lower your blood glucose. You understand? No? What is the end product of glycolysis? This is a pyruvate. Okay. When you have a high activity of glycolysis, as a result, glucose decrease finally. You understand? Could you please explain that?
طلاب هذا عندنا الجلوكوكاينيز يتميز عن الجلوكوكاينيز بانه لما ناكل كميه كبيره من الاكل، اوكي؟ هذا الجلوكوكاينيز يعمل كحساس او راصد. لما تدخل لي كميه كبيره من الاكل للجسم، هنا الجلوكوكاينيز راح يتفاعل، ولما يتفاعل راح البيتا سيل اللي موجوده بالبنكرياس تفرز لي الانسولين. الانسولين يحفز عمليه تحطيم الجلوكوز وتحويله الى بايروفيت، يحفز عمليه انتاج الطاقه. ليش؟ لان احنا ما يصير عندنا يكون الجلوكوز كميته كبيره بالجسم، هذا راح يؤدي الى هايبر جلايكوسيميا، يعني كثره الجلوكوز بالدم. لازم شنو؟ لازم تكون الجلوكوز نسبتها بالدم لا هي منخفضه ولا هي عاليه، لازم تكون متوسطه. تمام؟ فهنا عندنا شنو؟ لما يدخل الجلوكوز بكميه كبيره للجسم هنا البنكرياس راح يفرز لي الانسولين، الانسولين يشتغل كاكتيفيتور لعمليه التحلل الجلايكولايسيس. هنا بهاي الحاله الجلوكوز راح يكون عنده مسارين، اما يتحول الى جلايكوجين ويتخزن بالجسم او يتحول الى طاقه. وهذا الشيء احنا راح نقلل كميه الجلوكوز بالدم. واضح؟
And about the clinical point, maybe on our final exam, I asked about the enzyme that is deficient in one specific type of diabetes. And the name is a maturity onset diabetes of the young type, with the abbreviation of MADI. MADI2 is a type of diabetes, and in this situation, you have some alteration or mutation in the glucokinase activity. So, if I ask you which enzyme is deficient in MADI, it is a glucokinase. Do you know why? Because whenever glucokinase is manual, it cannot work. Even you actually eat a large cake, it sit down, okay? And the glucose reach to the 300. It says that it's none of my business, I'm manual, okay? It can result in a specific type of diabetes, but specific, as I mentioned, this is not diabetic 1, diabetic 2. This is a specific MADI 2.